Does anybody have any questions about anything? Movie content for today? Coursework? Anything at all? Please work on the Socrative quiz. Were you raising your hand? Go for it.
Do we have to do the puzzle your professor thing? Or was that part of the Common Genius thing?
Oh, yeah, what I had written on the syllabus as Puzzle the Professor was submitting questions for the mock exams. So people have been doing that the whole time. But hopefully everybody saw the announcement. The mock exam for the final is online, but that just covers the new material, like I said, not the old material that will also be on the final. And Wednesday we've got a review session. I will pass out more information about the format of the exam. It will be the same type of exam. It'll just be a little bit longer. I'll tell you how many questions and breakdown of how many points on previous material versus number of points on the new material that hasn't been tested yet on Wednesday. Yeah?
Will you be having office hours during dead days?
I will not have formal office hours during dead days, and I do have some random meetings scheduled here and there on Thursday and Friday, but in general, I'll be in my office. So if you've got a question and you're on campus, come knock, and if I'm in, I will open my door and we'll talk about genetics. Others?
Yeah, it must be.
Okay. So a good number of you came up with these Rube Goldberg machines. I just thought this was maybe an interesting, hopefully interesting way to close out thinking about how gene expression regulation works in prokaryotes or in eukaryotes. And the reason that I posted this sort of a question, make some sort of complicated contraption that turns on or turns off gene expression, is because in all honesty, this is kind of how complicated gene expression regulation is in the cell when you start thinking about something happens and another protein responds to that, and a protein responds to that, and a protein responds to that. Those of you that have taken biochemistry know about kinases, probably. That's kind of how gene expression regulation works. It's a huge series of proteins, one of which works on the next, and then there's a repressor, and then there's transcription, and then there's transcription of a transcription factor, and that transcription factor goes and tries to regulate gene expression somewhere. So it is like a Rube Goldberg machine, what's going on in our cells when genes get turned on and turned off. I'm going to load in a couple of your submissions. We'll take a look here. So what's going on here? This is the asthma example. So we've got pollutants. We've got some lead, apparently, and nitrous oxide being inhaled, going into the body, and somehow that lead increases T helper 2 cells. What did T helper 2 cells do? That causes asthma or not? Yeah, so this is a state of asthma. And in this case, we've then got NO2, this student or these students are suggesting negatively interacts with T regulatory cells. And those cells are the ones that inhibit T helper 2 cells to prevent asthma symptoms. So when you inhibit an inhibitor, it's a double negative. It's like NO2 also activates T helper 2 cells in a logical sense. So that's what's happening in this scheme. So we've got inhaled pollutants getting translated, not in terms of ribosomes. The effect is being translated or passed on to the T helper 2 cells. But my question still is, how does the entry of that chemical into our body do anything at all to gene expression? What's the connection between our environment and gene being turned on or gene being turned off? So how does this process down here work? Small molecule impacts gene expression. What have we seen in class where small molecule impacts gene expression? What example have we seen? Where something in the environment will turn on or turn off transcription. Like lactose, right? Small molecule. What does lactose do to affect gene expression?
Change the shapes of the juice.
Yeah, so lactose binds to a repressor molecule. There's an allosteric interaction. That small molecule sugar changes the shape of the repressor, and that affects gene expression. It's a fancy looking one. We've got basically the same idea. Here, though, the student has introduced the methyltransferase enzyme. That is the enzyme that's going to put methyl marks around the FOXP3 gene. So that's absolutely right. Methyltransferase is going to inhibit FOXP3. And what does FOXP3 do for us again? This gene increases Treg cells. So that's why these students have shown that this effect ultimately has a negative impact on those Treg cells. But I still have that same question. How does the binding of small molecules in the body how does that signal get passed on to the activation of this methyltransferase enzyme? Any thoughts? How do small molecules potentially affect gene expression? How what?
How fluids affect gene expression?
Yeah. And we know that they affect gene expression. Now the question is how? What's the mechanism?
So is it by causing cytokines to add a PhD group?
Yep.
And that prevents your binding factor or your...
Absolutely. So here's one way to think about this. We're looking at the expression of the FOXP3 gene. What's the situation in the cell where FOXP3 expression gets turned off in this case? What happens to the chromosome that turns off FOXP3 expression? Methylation. So up here in the promoter, there are going to be some CpG islands, right? C's and G's next to each other in the DNA sequence. They get methylated. Okay. What does that methylation? What enzyme? You better flip one or two slides back and
So it's not
look
the
it up.
FOXP3 that's getting methylated,
It's the DNA, it's the chromosome that gets methylated in this case. And that turns off gene expression of FOXP3. And there's an enzyme that does that. That's a methyltransferase. Because it transfers methyl groups. And basically every enzyme, if you ever wonder, has an ACE at the end of it. It's kind of what signifies that a protein is an enzyme. It ends in ASE, which apparently I can't write. So let me try that again. ASE, methyltransferase. Where does the methyltransferase come from? It's a protein, so how did it get made? Right, so that means that there is a gene. It's transcribed, it's translated. This protein gets produced, means there must be a methyltransferase gene somewhere.
That's probably upstream.
So I'm just going to, it doesn't matter where it is, I'm just going to diagram it on the same chromosome It doesn't have to, it could be to the left, it could be to the right, it doesn't matter. There's a methyltransferase gene. That's going to produce the enzyme that's going to go turn off FOXP3 expression. So how does the methyltransferase gene get regulated? How does its expression get turned on and turned off? Hypothesize with me. We don't know the answer to this, but take a guess. Let's say this is like the LAC system, and there's an operator. So what sort of a protein would we have to invent to regulate gene expression if there's an operator? A repressor. So let's say there's a repressor. And like LAC repressor, let's say that this repressor has an allosteric site, a site where a small molecule can bind and regulate the activity of this protein. In this scenario of asthma, what sort of small molecule might that be? Let's say, yeah, lead. Let's say it's a pollutant. What would the binding of the pollutant do to the repressor in this case, if this acted exactly like LAC repressor? Change the shape of the repressor. What would the repressor do? It would unbind. Right, so pollutant binds the methyltransferase repressor, let's say. The methyltransferase repressor lets go of the promoter. So now what can happen? Methyltransferase gets transcribed. So you get transcription of methyltransferase. You get the production of the methyltransferase enzyme. Remember, this is all in the presence of pollutants. So you get methyltransferase enzyme, and it goes down and methylates the promoter of FOXP3. You don't have any FOXP3 expression. You don't have any T helper cells or Treg cells. You generate T helper 2 cells. You get asthma. It's one possible way to think about how exposure to any sort of small molecule could mechanistically, physically that is, go from some substance in your lung to floating around your bloodstream. Some of you drew pictures of particles going in the lungs, getting into the bloodstream, getting bound by antigen-presenting cells. But ultimately, this was trying to review one way that you might think about gene expression regulation. Small molecule changes protein shape, affects gene expression. That's not the only way gene expression regulation happens, of course. We've talked about the GAL system as well, and that's a little bit different. But that also involves binding of a small molecule as well. And that's often how organisms respond to their environments. So that's just one example of how a small molecule could affect gene expression. All right. That should have given us sufficient time to collect all of your knowledge about this Socrative activity. Let's check out your answers. Okay. Oh, maybe there are 3 of you still figuring out sex of an XX human. That's fine. Let's see. Vastly female. Good. You've been paying attention. So XX is female in humans. Why is that? Somebody tell me in 15 seconds or less.
Females are superior.
Because females are superior. It's a very, you're going to be a politician someday, aren't you? Okay, so in humans, the absence of a Y, the Y chromosome is what decides. If you have a Y, you're male. If you don't have a Y, you're female. And we're talking about physiological sex. I'm not talking about sociology or anything like that. So let's see. How about XY fruit fly? Male. For those of you that got it right, XY fruit fly is male. What is it about fruit flies and sex chromosomes that makes you arrive at that? What controls sex determination in fruit flies? It's the number of X chromosomes. So it's not the Y chromosome. Y chromosome has nothing to do with sex in Drosophila. In humans, yes. Fruit flies, no. So there's a similarity, but a difference between fruit flies and humans. So what's the similarity? They have two genders. They have males and females. So that's the difference.
Well, it is represented with the same letters.
Right. So in humans and fruit flies, if you have two X chromosomes, you're female. That's the similarity. If you're XY, you're male. What's the difference? Which one does the sex determination? The X chromosome is the sex determining element in Drosophila. It's the number of X's. In humans, it's the Y chromosome that tells you whether or not you're going to be male or female. Hence, the next two questions, which I'm expecting a bit more variety. And maybe I didn't word this question entirely well, because the option of hermaphrodite may be interpreted differently by many of you when you choose it as an answer. So let's just go male versus female. Seems like most people think male, and not so many think female. So with the knowledge that the Y chromosome in humans controls sex determination, is it going to be male or female? It would be male because presence of the Y chromosome in humans mainly determines sex. So yes, it's true, Klinefelter's individuals, XXY individuals, do have some feminine features as well. So they get gynecomastia, they get kind of enlargement of the breasts, but they still have a penis, but sometimes they're sterile. So hermaphrodite's a valid answer in that they do develop two different sexes, tissues at the same time. But in general, the point is they're more male than female. And that's because the presence of the Y chromosome enables that development of at least some male traits. So what about XXY fruit flies? What controls sex determination in fruit flies? It's the number of X chromosomes. So if you've got XXY, you've got two X's. Whoa, almost tied. So female here. XXY fruit flies are female. Like Klinefelter's humans, they're often sterile or have other problems with reproducing, but by and large, they look like female Drosophila. Even though they have a Y chromosome, because in Drosophila, the Y chromosome has nothing to do with sex. It's just there. It's not like the Y chromosome in humans, where that drives maleness. In Drosophila, doesn't matter. In fact, let's try this. What happens if you're an XO Drosophila? You have an X chromosome and no second copy, no Y or no second X. What's your sex? Male, because there's only one X, which equals male. The Y chromosome does nothing for, practically nothing for Drosophila. It's just there. Weird. Chromosome that, it does have some genes on it. It does do some things for the males, but it's not critical for sex determination. What about an exo human? No Y chromosome, no male. So that's Turner syndrome, and female. Or female-ish, at least. No development of male genitalia, testes, anything like that. Quick review from the movie for today on sex determination. Does anybody have any questions, concerns, details that I've omitted you'd like to know about? Bless you. One of the movies that got posted on our box folder over the weekend, which I do encourage, as always, all of you to go take a look at, mentioned small inhibitory RNA molecules. In last class, one of you brought up this concept of RNAi. It's in the news a lot, so I thought it would be a useful thing to talk about briefly in class. So it's a method by which humans make transgenic organisms. It's a method by which we affect gene expression in organisms. Has anybody here heard anything about RNAi before? What sort of knowledge do we have in the class already about RNAi?
Isn't it when they make like fly stills in agriculture, so it can't reproduce, and then they don't have an infestation and they're pests?
So you can use RNAi, it's a technique, to do a lot of things like make animals sterile. You can do it pretty much anything you want with RNAi. Well, we'll talk about a few limits.
It activates a gene to turn on. Like you can use it in plants to spray, and it activates an anti-fungal.
Okay, so RNAi is used to turn genes off. Now, if you turn off an inhibitor of something, then you activate it. So you can do things in plants like spray them with this compound, which we'll talk about, and turn off a repressor or something that turns on a gene that you're interested in. So RNAi, the reason we talk about it in genetics is because the first three letters, RNA. RNAi uses short molecules of RNA. Some of these are naturally occurring. So we have genes in our bodies, and other organisms do as well, that don't produce proteins. We've talked about this before. What are some of the types of RNA genes that our body has that don't get translated? They don't make proteins. Their goal, their purpose is to be RNA. tRNA, ribosomal RNA are a couple of examples. So RNAs, that's their function in the cell, is to be RNA molecules. There are genes that produce short RNA molecules that form hairpins. Hairpins. So that's one single-stranded RNA molecule that binds to itself and creates this structure. And the way, let's see, where are the RNA molecules in the cell that produce hairpins that we want to have? Which of these two molecules on the right, tRNAs and rRNAs, normally produce hairpins? They bind to themselves. tRNAs definitely. Ribosomal RNAs do too to a certain extent. So those molecules also form hairpins. Where does translation happen in the cell? By the ribosome in the cytoplasm. Right, so remember, messenger RNAs and eukaryotes have to get out of the nucleus to be translated. So where does our cell expect to find double-stranded RNA molecules? In the cytoplasm where the tRNAs are, where translation's happening. But our cells don't expect to see double-stranded RNAs in the nucleus. And our cells have surveillance machinery, enzymes, that float around looking for double-stranded RNA. Why? They're trying to look for double-stranded RNA and get rid of it. Because some viruses have double-stranded RNA genomes, and so our cells have this defense mechanism we have enzymes that float around in our cells looking for double-stranded DNA like hairpins, these short RNA molecules that form by themselves double-stranded RNA. And that's because that's the first step in the recognition of, hey, maybe my cell, the cell I'm in, this enzyme thinks to itself, has been infected by a virus. So the detection of double-stranded RNA then sets off a cascade of events we're going to talk about based on this slide. So the idea here is the top half is what's happening in the nucleus. The bottom half is what's happening in the cytoplasm. So you see there's a diagram of a hairpin. And here's a protein that's involved in recognizing that double-stranded RNA molecule. In Drosophila, these are all names for Drosophila, although they often get applied, the names of these proteins, to other organisms as well. Drosha is a protein that binds double-stranded RNA molecules and exports them or leads to their export out of the nucleus into the cytoplasm, where this fantastic enzyme called dicer, guess what it does? Dices. Dicer binds those double-stranded RNA molecules, and it actually does cut that double-stranded RNA molecule. And that's why you see, for example, before dicer binds and after dicer binds, the hairpin is gone. So dicer actually physically cuts parts of this double-stranded RNA molecule, and then that double-stranded double helix that's left binds to an enzyme complex that contains one protein called AGO2, Argonaute 2. This protein complex, RISC, stands for RNA-induced silencing complex. And that silencing part should give you some hint about what this is going to do. It silences or turns off gene expression of a gene that matches whatever the sequence of this RNA molecule is. So in other words, the sequence of this short RNA, which sometimes is like 20 or 30 nucleotides long, it is 20 or 30 nucleotides long. So it's a little bit longer than is diagrammed up here. The sequence of that RNA molecule is identical to a gene that's present in the genome that's producing transcripts. So there will be other molecules around here out in the cytoplasm being translated, shown, for example, right there at the bottom. The messenger RNA with its 5' end on the left and 3' on the right has a sequence that's identical here to the sequence of that RNA molecule. So after dicer works, that double-stranded molecule that's here called an siRNA, that's short inhibitory RNA. That double-stranded double helix of RNA gets bound by the RISC complex. And what that RISC complex does is kind of like DNA replication all over again in a very, very, very broad sense. That protein binds the RNA molecule, and it uses it, that short inhibitory RNA, as a template. That protein and its single-stranded RNA molecule go around basically checking out all of the transcripts, those single-stranded mRNA molecules in the cell, looking for a base pair match, which is what's diagrammed at the bottom. So when the RISC complex with its single-stranded RNA template find a match down here on the bottom, that single-stranded RNA template has base paired with a messenger RNA. Then the RISC complex shoots that messenger RNA off for degradation, elimination. That gets cut up into little tiny pieces and never gets transcribed or translated. It's already been transcribed. So that's how RNA interference or RNA silencing works. The short RNA molecule whose sequence is complementary to a gene. And dicer acts on the hairpin to get rid of the hairpin, producing a double-stranded template. That double-stranded template gets bound by the Argonaut protein in the RISC RNA-induced silencing complex. And then that complex goes around looking for transcripts that contain that RNA sequence, gets rid of them. And so all we did as scientists was take advantage of the fact that our cells have this system in them, and plant cells have this system in them, and other mammals, vertebrates, etc. Not bacteria. We take advantage of that. How? What can we do to use this system that's in the cells of organisms that naturally gets used to turn off gene expression?
You just insert little double-stranded RNA molecules.
Right. We create those double-stranded RNA molecules. They're short, they're tiny, they're cheap to synthesize. You pay a company to make these little hairpin 20 to 30 nucleotide long RNA molecules. You pay the company in Iowa to do it. They ship it to you. You inject them into cells, and then that process turns off the expression of whatever gene it was you synthesized this RNA molecule to be complementary to. So it's one way to turn off expression of certain genes. This is not by regulating the initiation of transcription. This is by actually, after transcription of the gene happens, getting rid of all the messenger RNA molecules that contain that gene. So here's a quick example. Told you before, so this brief review, 2 sexes in Caenorhabditis. The point here is that only the hermaphrodites make oocytes. Males only make sperm, hermaphrodites make sperm and eggs. So a normal hermaphrodite, if you look at it, if you look at a picture of the hermaphrodite, you'll see this little tiny spot where there are lots of little teeny tiny sperm. Sperm are minuscule like they are in humans compared to the size of the oocytes. And you'll see all these huge fat primary oocytes lined up in the gonad. So you get big cells, you get little tiny cells. The big cells are the oocytes, the tiny cells are the sperm. Well, we know something about how sex determination works in Caenorhabditis. So if you want to, this is C. elegans, for example, the best studied Caenorhabditis species. So let's say this is C. elegans. Let's say you want to make a female C. elegans. What are you trying to do to a hermaphrodite if you want to turn it into a female? You take away the sperm. You're trying to make a worm, a hermaphrodite that doesn't produce its own sperm. So we try to make a spermless hermaphrodite. Well, there are genes that you can target with short inhibitory RNA, the RNAi pathway, that turn off spermatogenesis. So we know the genes that turn on spermatogenesis. So what happens if we inject a hermaphrodite with an RNAi that prevents the expression of that gene that turns on spermatogenesis? We get no sperm production. We produce, instead of a hermaphrodite, a female. And that's exactly what happens. In this case, it was the opposite, actually. We injected a RNA molecule that was complementary to this gene called GOLD1. GOLD1 is important instead, in this case, for oocyte production. So remember how I said the oocytes are these huge, fat cells. Up here, each of these things that's labeled with an O is an oocyte. So there's one oocyte, there's a second oocyte, there's a third oocyte. They're huge cells. That's the width of the body of the organism, by the way, so they're half the width of the worm. That's a wild-type hermaphrodite. And then you compare that to what happens when you inject this worm with GOLD1 RNAi. So it turns off oocyte production. So now we have a gonad that doesn't have, this is an adult again, but it doesn't have any of those huge oocytes. It has lots of little teeny tiny sperm. And because I know you're not Caenorhabditis anatomy experts and you're just looking at this big gray blob and saying, yeah, sure, whatever, which is what I did before I started working with these organisms too. You can also do something to visualize the nuclei. So here we're looking at DNA, which maybe makes it a little bit easier to see. That there are lots, in this mutant down here on the bottom, there are lots of little punctate bright dots. Those are nuclei of sperm. So every one of those bright little dots, we stain this with DAPI, which is a chemical that binds to DNA and fluoresces. So you can easily see the nuclei of all the cells. And there's a clear difference between what's happening on the top here, where there's a huge oocyte. There's no fluorescence in here except for little tiny dots. That's the nucleus of the oocyte right there with chromosomes. And these few little dots down there, those are sperm that are smushed up between the oocyte and the inside of the gonad. So they're basically sitting on top of the oocyte as we're looking at it top down like this. But most of the sperm here are surrounding this oocyte. So all those little tiny dots kind of looks like a disco ball or something. That's sperm surrounding this oocyte after it was fertilized. So the first sperm got in, the oocyte says, oh, no more. And then all the rest of the sperm just kind of are smashed around this oocyte in three-dimensional space. But on the bottom, simply by microinjecting, you take a needle, fill it with liquid that contains this double-stranded RNA molecule, you stick it into the gonad of the worm. Yeah, I know, it's not great for the worm, but they survive. And their offspring don't produce oocytes. So this is a hermaphrodite. It's got a vulva, like a normal hermaphrodite would. Its germline has been changed. We've altered its germline not to produce its own eggs in this case, which is great, for example, if you want to do what? Be creative. What's something that you could actually use this for in the real world?
Kill off a generation.
Kill off. So how would you use this to kill, eliminate, make extinct?
You can produce an entire generation of just one sex.
Right. So this could be used for biocontrol. If you found a way to go out into nature and microinject every worm of this species with this RNA, which can't. But that's where this whole idea about spraying and vectors for getting RNA molecules into organisms comes into play. But stepping back for a second, we can't go inject all the worms. But if you could, you would create a bunch of once hermaphrodites that are now just producing sperm. And if all you have in your species are sperm producers, then there is no next generation. So you've killed the species. So you could potentially use this for biocontrol, and many other things as well. That's just one example. So then the tricky thing is, most recently people trying to figure out, can you spray crops with maybe a virus or something to get into plants that would deliver a double-stranded RNA payload into the plants to affect gene expression. That's sort of the current state of affairs in terms of bioengineering with RNA.
RNAi represents the gene that produces sperm. Is there a counteract to that? Like C. elegans can make sperm?
So in this case, we designed this small RNA that we injected to get rid of oocytes. You can also do the same thing with sperm. So we know the genes that are responsible. This is incredibly important. You can only RNAi. It's only useful when you know the gene that makes the phenotype that you're interested in. So you have to know something about the genes. So because we know what genes make sperm in C. elegans and what genes make oocytes, you could make a different RNA molecule that would get rid of the sperm instead of getting rid of the eggs. In this case, that would have the same effect on a hermaphrodite. You either have one that makes only sperm, or you have one that makes only eggs. Either way, by itself, that individual can't reproduce. The problem is you have to get every single individual in the population to be the same sex, or else there's going to be some reproduction, and and you haven't done anything, other than made some really cool worms. Yeah?
What if you turn off both the oocytes and sperm?
Right, you said it again, since, thank you. Okay.
What if you turn off both the oocytes and sperm?
Ooh, what if you turn off sex determination altogether, or germline production? Then you get a sterile worm that doesn't produce any gonad, or any germline cells. That's even better, right? For biocontrol anyway. Not for the worm. But yeah, you could do that too.
Along that same idea, there was a study where they changed the gene in male mosquitoes so that they require this specific kind of sugar that is obviously available in a lab, not in nature. So they make a bunch of these mosquitoes and release them out in the wild. They go out, have the next generation of mosquito that is reliant on the sugar. Obviously, it's not in nature. And now you killed off a generation of mosquitoes.
Right. So you can do lots of biocontrol with this. Then the question gets to, do we want to be releasing transgenic mosquitoes, for example, into nature to wipe out a population of mosquitoes? Well, yeah, on the one hand, controlling disease, so mosquitoes are vectors for a lot of diseases, so if you can find a way to kill them, and that's why they did these sorts of things. Try to, prevent malaria transmission, other sorts of diseases. But as our GMO expert, for those of you that went to his talk, discussed a couple Fridays ago, what happens if that mosquito escapes and goes and starts reproducing with other populations that aren't, or other species, that aren't vectors of diseases? Then we're wiping out more species than we originally intended. And then you have to start worrying about, how far does this spread, because it's hard to control. So, we all have to weigh the good and the bad. I'm going to leave this to you to think about. So we're almost out of time. Benefits and costs, or pros and cons of using RNAi versus using something like a gene deletion. So you could make a worm where you just make it sterile by microinjecting it with double-stranded RNA, and only in that worm it doesn't produce one type of gamete, sperm, say. Or you can create a worm that doesn't have the gene at all in the first place that's necessary for it to make sperm. So it's useful to think about why we use, in genetics, both strategies. There are some situations where one's useful, some in the other. Yeah.
But don't, proponents of RNAi say it's useful because you can turn it on and off by application level? So if you quit applying the RNA to the species, then eventually they'll start producing their RNA and it'll be
Right.
expressed again.
So that's one benefit, if you want to think about it as a benefit, of RNAi, is that you have to microinject the worm whose offspring it is you want to show that trait. But once that's done, the lack of oocytes, for example, the lack of sperm in this example, doesn't pass on from generation to generation. You've just affected one generation of worms, individuals, not the whole lineage. That's a pro for biocontrol, because that means you don't have to worry about the effect escaping and getting passed on generation to generation from worm to worm, because that doesn't happen with RNAi. But that's also where this RNA versus gene deletion comes into play, because that also means that if you want to see the effect in multiple worms, you have to inject these microscopic animals, these little tiny needles, every time you want to do an experiment or do something with them. So that's the pro-con, it's a trade-off. RNAi is not long-lasting, which is great in some circumstances and horrible in other circumstances. Okay, so I need to launch this new Socrative activity. And while I'm working on that, I'm going to tell you this is not something you have to do, although I do appreciate your feedback again. It's another survey. This is the one where I ask you if you have advice for future students that are going to be taking this class from me in fall. So if you want to, I'm going to leave this open all afternoon, so you don't have to do this right now. But if you do want to leave feedback, I will pass it all on to next semester's students. just a second. And then I'm going to show you. So this is all anonymous. You'll note I'm not asking for names. I'm not asking for student ID numbers. Last couple points. So next class, we've got a review session. Bring questions as normal. The whole 15 minutes is for me to answer questions that you have. Group notes, remember, are being graded for the last time on Wednesday. Everything is due on Wednesday. So we currently have 3 term-end surveys with URLs on Blackboard. I've posted one each day. Today is a new one, aside from this Socrative activity. So if you can please do those, again, by Wednesday, end of the day, 11:59 p.m. Videos for Comedy Genius or Genetics and Society. Please go watch them and leave comments. There will be a fantastic prize on Wednesday for the video that gets the most comments. I noticed some of you that posted videos are commenting on your own videos, which is legit. That's fine. Vote for yourself. That's good. And please send me your presentation file. For those of you that have done the in-class presentations this semester, if you didn't send me your file, please do. And here's that set of 3 surveys on Blackboard. Those 3. 1, 2, 3. All due on Wednesday. See you for our last class in 2 days.